very pleased to introduce to you Dr. Richard Frye. He's an outstanding physician and autism researcher. He is a child neurologist. He has been the author of over 200 research papers. He's been involved in many clinical trials. I'm very pleased to have had the chance to work with him on several studies, including our work together on the Microbiota Transplant for adults with autism. We received $1 million grant from the federal government to do that research. So very excited for him to tell you about his work, to introduce him to tell you about his work on mitochondria. He's really the expert on mitochondria function in autism. Please join me in welcoming Dr. Frye.
Thank you so much for that great introduction. Today I'm going to talk about treatment of gut problems in autism and how treatment that Dr. Adams has developed. Really, we've found that supercharges the body's powerhouse called the mitochondria. However, I'll take you through a little tour of how we think that does it and things about the microbiome, to try and be complete. First, I always have my disclaimer, which just says, any type of treatments we talk about here are not FDA approved, so they need to be prescribed or done by somebody that knows the risks and benefits of any treatments. So I'm not endorsing them medically in any way, but you guys probably know that. So we know autism is a multifaceted disorder, and I have this slide that really shows all the different parts and pieces of abnormalities that we see in autism, and it's really kind of overwhelming when we think about everything that's going on. We have our environmental factors, which you see on the right. Many different environmental factors, including psychosocial factors to environmental toxins. On the left, we have these systemic disorders. So things that don't only affect the brain, or any organ in particular, but affect the whole body. One of those is the mitochondria. The mitochondria is the powerhouse of the cell and is in almost every cell of your body, and it's needed to make energy, among other things. And then in the middle, we have many of the organs that are dysfunctional, including the brain, which we think about a lot. But what we found is this connection between the brain and other parts of the body, including the immune system and the gastrointestinal system, where we see the microbiome. So in recent decades, we've been learning more and more about the microbiome. We're learning that really the microbiome, which are these organisms that live in and on our body, throughout our body, help us in many ways for our body to work. And it's in very complex ways. And so we're just learning more and more about them and how they can cause disease and and promote health. One of the things that we're learning is that some of the things that we do actually can disturb the microbiome. So you actually inherit your microbiome from your mother at delivery. And that's just assuming a normal delivery. However, we've changed our practices and we do things like do C-sections, so the baby doesn't get the bacteria it should. We many times give antibiotics, and that kills the microbiome in certain species, so it can disrupt it. Instead of using natural breastfeeding, we use formula feeding, which doesn't have some of the natural products in breastfeeding that actually stimulate the microbiome to develop. And then really the quality of our food is sometimes will disrupt what balance we have in the microbiome, because it can disturb which bacteria actually live. And so we've thought about in recent years that there's this this thing that we see that comes together where having an imbalanced microbiome combined with genetics and environmental insults can really destabilize our immune system and cause many autoimmune diseases, we believe. At least there's some theory there that we've lost some of our key bacteria or other organisms that stabilize our microbiome, and it may be related to the rise in autoimmune diseases and other things that we see. And one of the real major roles in the development of our immune system early on, and our metabolic systems, as we'll talk about in a second, is the microbiome starting from birth. And if that's depleted and doesn't give the body the right signals, we can run into problems as far as development. Many of the things that we see with the microbiome depletion can be combined with environmental toxins such as acetaminophen and other things like organophosphates and such to cause inflammation, which causes metabolic disturbance and oxidative stress. And these abnormalities in inflammation, metabolic dysfunction, oxidative stress is something we're seeing in many different diseases, but particularly autism, where we see them in many other metabolic and other types of diseases, such as diabetes and such that are rising, in our population. And so one of the things is we're learning that the microbiome is so important to stabilize these systems in our body. And this is just some of the examples of things that have been linked to abnormalities in the microbiome, including autism, psychiatric disorders such as depression, anxiety, asthma, and other atopic diseases like eczema and such, cancers such as colon cancer, hypertension and heart disease, obesity, metabolic disorders, inflammatory bowel disease, and peripheral vascular disease. So this microbiome is so important, and we've linked it to many diseases. And now, as we learn more, we can learn how to maybe solve these diseases by manipulating the microbiome. So how does our microbiome influence our body? Well, there's many different ways. These bacteria do several different things. One of the things that's very important is they regulate the immune system. So 70% of our immune system is in our gut, and these bacteria will actually stimulate the immune system, and that will affect the rest of our body. The bacteria can help certain cells that make neurotransmitters function so that it can affect neurotransmitters that go to the brain, including also fatty acids. So many of the nutrients that we get go through the microbiome, and because of that, depending on our microbiome, we may or may not get the optimal nutrients. And that's important for brain growth, including vitamins like folate, also neurotransmitters, and certain types of fatty acids. Our brain is really made of fats and fatty acids, and if we do not get those nutrients, then it will not allow the brain to develop optimally. Also, the microbiome stimulates certain nerves. We have something called the vagus nerve, which connects the gut to the brain. And so we can actually transmit signals to the brain from the gut. So it's important for the gut to be healthy in order to stimulate the brain correctly and send the appropriate signals. Here we can say this is more a diagram that shows how certain types of bacteria can influence these cytokine production, which are the inflammatory mediators that tell our immune system to work, and other types of what we call regulatory cells. And we have something called the GALT, which is really the immune system that's in the gut. And so all of these bacteria can really affect how our immune system develops. One of the other things that we're going to talk about is how the microbiome affects metabolism. And metabolism is really defined about how our body gets its fuels, gets rid of its wastes, and makes the nutrients that it needs. So some of the major metabolites that the bacteria make in our microbiome are called short-chain fatty acids. And how they're made is that a lot of dietary fibers that we eat go down to the lower gut, and they ferment. And when they ferment, those bacteria make these short-chain fatty acids that affect metabolism. And really the major ones that we see are acetate, propionate, and butyrate. And here, another diagram. So fiber-rich foods go down to the microbiome, make these short-chain fatty acids. One of the things, the major effects that we see is that some of the short-chain fatty acids affect insulin production and signals through leptin for body weight. So we know these short-chain fatty acids can influence insulin production, diabetes, and also result in obesity and preventing obesity if you have the right fermenters. The short-chain fatty acids have a wide range of other effects in our body. They actually influence the integrity of the intestine. In fact, butyrate is a fuel that the intestine uses, the intestinal lining. In mice, it's actually the only fuel it can use. So if you don't have butyrate produces in mice, the intestinal lining won't be healthy. It affects wide other parts of the body, such as the lungs, and can regulate and decrease inflammation in the lungs. The pancreas, and can regulate insulin secretion, which can help control blood glucose, affect adipose tissue and breaking down of adipose tissue, regulate hemopoiesis, the development of blood cells, regulate immune cells and decrease inflammation. In the brain, it can not only affect satiety for roughly food intake, but it actually regulates neurogenesis, how the brain and how the neurons grow and develop, and the microglial cells, which are the inflammatory cells of the brain. In the muscle, it stimulates the energy in the muscle, regulating energy storage and mitochondrial content. So the mitochondrial is the energy, the powerhouse of the cell, and you need many of them in the muscles for your muscles to be strong. In the liver, many different pathways for lipid storage, for insulin sensitivity, and then it also affects the enteric nervous system, the nervous system that regulates the gut and changes things like gut motility. Again, metabolites have many different effects, and some of the effects also are within the gut to regulate proliferation of the colonic cells, control colonic inflammation, and regulate colonic tumorigenesis. So actually, butyrate there's some evidence that it can prevent colon cancer. So depending on how you actually produce these short-chain fatty acids, you can actually prevent cancer and regulate the health of your gut. What's interesting is that these different short-chain fatty acids change depending on the time, the age. In early stages of life, you have different distributions of gut bacteria, particularly because of being breastfed. Feeding produces human milk oligosaccharides, but are very specific to actually feed bacteria that will make the proper short chain fatty acids. Usually acetate is the major one that's produced early stages in life. And as we become an adult, we have a shift in our bacteria to increase firmicutes and bacteroides, and we make propionic acid and butyrate, which are different short chain fatty acids and have different purposes. And then as in the elderly, we see actually a decrease in the microbiome diversity and a decrease in the short chain fatty acids, which probably causes some of the issues with GI issues in the elderly. So, not all of these short chain fatty acids are the same. So I've been talking about how many of them do really great things. Well, some yes and some no. Butyrate tends to be a very good short chain fatty acid, and we can look over here of some of the things that it does. It improves insulin sensitivity and insulin response to glucose. But propionate can have slightly different effects and actually can increase the risk of type 2 diabetes. So, so not all fatty acids are the same, and we'll see here that we have a kind of balance between some of the good and not so good fatty acids, butyrate versus propionate. And so propionate really has been linked to autism, and this is really some work that has been done in about a decade ago, where a lot of experiments were done on propionate to actually look at how it can actually cause autistic symptoms. And there is a model of autism actually produced by using propionate. So this short chain fatty acid has all types of effects, and some of them aren't all that good. It can cause neuroinflammation and problems with neurodevelopment. It can alter mitochondrial function, we'll talk about. We have models that show that it actually causes autistic-like behaviors. It can change gene expression and cause inflammation in the gut. So about 15 years ago or so, a researcher named Derek McFabe actually produced a model of the propionate model of autism in rats. And what he did was inject the rats' brains with propionate. And what he found is, and this is in adult rats, he actually found that the rats transiently became autistic, these adult rats. And so these are some of the examples. What he did is he followed and he looked at the rat's behavior. And so you can see what he did is he put the rats in these round cylinders and watched their behavior. And then measure their behavior. And some of the things that he found is that when he injected propionic acid, he found that the rats had a higher total distance of running around. So this is akin to hyperactivity. So instead of going around and exploring, they actually ran around and around and around in this box. They also had repetitive movements that look like stereotypies, which we see in autism. And the ones up here with propionic acid had a higher number of these repetitive movements. The time that they spent moving too was increased. One of the really interesting thing was a social experiment. And what they would do is put a rat in the middle here, so this is a novel rat, and this and on the left you see the rat that was treated with propionic acid, and what you see is that this rat is running around the outside of the cylinder, and really doesn't spend a lot of time at this new rat to say hello, smell them, investigate them. Whereas the one next to this is the where it says PBS is the rat that was given the placebo instead of propionic acid. And you can see the difference. It would circle this novel rat and smell it and investigate it and be social. So just by injecting this propionic acid, the social behavior of this animal has changed. And this can be measured with looking at the proximity, the duration and proximity to the novel rat. And we can see that in the black bar, it's decreased in those that were given propionic acid. And there's, if you bear with me, it's really impressive. There's some movies we have of this. So we'll look at the rats the placebo rats. And so you can see here, this is a rat that's not treated with propionic acid. And what we can see is that this rat outside that wasn't treated is exploring this novel rat and that's in the little cage. He wants to say hello to him. He wants to smell him. He wants to investigate him. And he's showing normal social behaviors. He's circling that novel rat to really see, to make friends or find out what that rat is about. Those are social behaviors. So then we look at the rat that was given propionic acid. And you can see the striking difference. The rat that was given propionic acid doesn't even recognize that there's a new rat in there, somebody new to say hello to, just circles the chamber around and around and ignores the fact that there actually is any other rat to socialize with. Right, so we saw that the social ignoring signal. And what's really interesting is when they looked at the rat's brain after they gave propionic acid, they stained for these things called microglial cells, which are inflammatory cells in the brain. And you can see on the left where it says placebo, there's no inflammatory cells. Inflammatory cells are these brown dots that you see on the slide for propionic acid. This is actually a slide of the brain. And you could see that the rat that was given propionic acid has all of these brown dots, which are these microglial cells or inflammatory cells of the brain. And so we're learning that that propionic acid, not so good, but butyrate, another short chain fatty acid, has actually very beneficial effects and actually helps decrease dysbiosis, improves liver metabolism, decreases obesity and food intake, and also regulates as an anti-inflammatory cell, for anti-inflammatory signal. So the other thing, besides being signaling molecules, these also affect something called the mitochondria. So the mitochondria, or as you see, the mitochondria, because it's very mighty, is the powerhouse of the cell. Each cell has several mitochondria. That is, your cell has anywhere from hundreds to tens of thousands of mitochondria for how much energy it needs. And you can see here it's circled in red inside this basic cell. One of the unique things about the mitochondria is that it has its own DNA. So the only other part of the cell that has DNA is in the nucleus, and that's where the chromosomes are. And that's where you always think of where your genes come from. But it ends up that the mitochondria has its own DNA to actually program itself. And that DNA, the mitochondria, and the DNA in the mitochondria are inherited from your mother. So it's not like normal inheritance where you get one half from your mom and one half from your dad. You actually inherit your mitochondria from your mother. So the mitochondria is a very complex, what we call organelle. It has a outer membrane, which gives it shape like a bean, and it has its inner membrane, which has these folds. And these folds, which are called cristae, are very, very important because that's where the energy is made inside here. And inside what we call the matrix is where the DNA is and where many of the different machinery is to actually make the proteins and such that need to work to make energy. And so as a simple example, we eat fats and sugar, and they go into the mitochondria, and they're brought into this thing called the citric acid cycle. And as the citric acid cycle spins, it takes our fats and sugar and makes it into these things called electron carriers, which carry energy over to something called the electron transport chain. And this is the electron transport chain. It's called a chain because it has a number of different enzymes, or you can think of them as little machines that process these carriers as we go along. And we go from 1 to 5. And at 5, number 5 makes something called ATP. And ATP is the energy molecule of the cell. And then that goes out of the mitochondria and gives us all our energy. The mitochondria is extremely important because you can think of all of the parts of our body that need a lot of energy. Your heart is always beating. It needs a lot of energy. Your nervous system is particularly dependent on the mitochondria. It uses, despite the fact that it's only a small percentage of your body's weight, it actually uses about 20% of the oxygen and energy, and makes about 20% of the energy in your body. Your immune system is very energetic. The immune cells are moving all around your body. They're attacking invaders all the time, trying to protect you. The GI tract tends to be a place that needs a lot of energy too, because it's doing a lot of different things. It's moving around your food all the way down this very long pipe. It's digesting your food. And of course, your muscles. They need energy to work also. So when we look inside the mitochondria, we can see that it's extremely complex. And this is just kind of a sketch of some of the pathways inside the mitochondria. And I'm not putting this up here for you to kind of memorize it or kind of understand it, but to make the impression that it's a very, very complex organ. And this is the machine that is important for regulating all of the fuels that come into your body and making all the energy for your body. What we're learning is that mitochondrial dysfunction really is seen in many, many, many kids with autism. We know that there's two types of mitochondrial problems. There's something called mitochondrial disease, which is thought to be rare, and is a state where the mitochondria is working very, very, very poorly, and many times causes more of a degenerative course where you become sicker and sicker. This is thought to be a rare disease actually in the general population, but we know about 5% of children with autism have a mitochondrial disease, can be diagnosed mitochondrial disease. However, there's something else called mitochondrial dysfunction, and this is when the mitochondria isn't so sick that it doesn't work at all, but it's not working as well as it should, and so it can't keep up to help the body do its perform its normal function. And that's sometimes, that's okay if you tend maybe to be a lazy person, but it's not okay when your body is growing and your brain's developing. So when you have mitochondrial dysfunction in the state of a growing body and growing brain, those things can't develop because they don't have the energy to develop and they stall out. And we're finding that many children with autism, maybe up to 80%, have a problem with mitochondrial function. So this is a sketch of the mitochondria and how and some of the pathways that we have. And so again, I'll just give a brief overview. We have glucose down here, which is our sugar that comes in, and it's turned into something called pyruvate and outside the mitochondria. And then pyruvate is the major thing, the major molecule that goes into the cell and starts to make energy. One of the things that we see if you've ever had an investigation of your mitochondria, your child's mitochondria, you know that we measure certain things. And some of the things that we measure are elevations in lactate, pyruvate, and also the amino acid alanine. And the reason why we do that is that's the entrance to the mitochondria. So if the mitochondria isn't working, we get a backup, and these things actually get elevated. On the side here, we also have fatty acids come into the mitochondria. And carnitine is a very, very important vitamin that's needed to pull those and shuttle those into the mitochondria. Once inside the mitochondria, we go into this thing called the citric acid cycle, the TCA cycle, the Krebs cycle, many names for the same thing. And this is the cycle that goes round and round, and we produce all these different intermediate metabolites, which help us make energy. And when we look for mitochondrial dysfunction, we also measure all these different metabolites to see where there might be some type of block in the citric acid cycle here. Again, the citric acid cycle goes around and makes these things called energy carriers, NADH, FADH2, and they go to the electron transport chain here. Now, the electron transport chain, again, is this series of 5 little machines that help us make energy. We can see that one of these carriers, NADH, goes to complex 1 here, and FADH2 goes to complex 2. And then something called CoQ10 carries the energy from complex 1 or complex 2 over to complex 3. So this is very important because a lot of people wonder why we say to take CoQ10, or in its reduced form, it's called ubiquinol, when you have mitochondrial problems. And this is why, because ubiquinol, it's thought of as an antioxidant, but it's not just an antioxidant, it's actually the wires that hold this electron transport chain together. Once we get to complex 3, we go to complex 4. Complex 4 is where we use oxygen. So this is actually the whole reason we breathe is to get oxygen to complex 4 so we can make energy. And then complex 5 makes this thing called ATP, which goes to the rest of the body and the rest of the cell and makes our energy. The other thing to notice is that fatty acids come in over here, and they go right into complex 2. So when we look at these different parts of the electron transport chain, we can look at this in a fashion of complex 1 tends to process carbohydrates, complex 2 tends to process fats. So we can know, depending on what abnormalities we might see, which fuel might be better for the mitochondria. So here's just another picture of what we were talking about, this Krebs cycle, citric acid cycle, or tricarboxylic acid cycle, goes around and makes these energy carriers that go to the electron transport chain. So why am I telling you this? Because now we'll go back to these short chain fatty acids. These short chain fatty acids have a role that's very integral to metabolism. And so as we were talking about before, we have butyrate. Well, what does butyrate do? Butyrate comes in to the top here into the citric acid cycle and goes in just like pyruvate and goes around and makes energy. So that's a really good thing. But what about propionic acid? We were talking about this other one. Well, propionic acid actually, instead of going into the citric acid cycle, actually steals one of the first metabolites called acetyl-CoA and pulls it out and has its own kind of separate side metabolism that goes down the side here and then comes back in to the citric acid cycle down here. And so what does that do? Well, the citric acid cycle was making these energy carriers, NAD. And you can see what happens is we're actually bypassing that part. And now the citric acid cycle can't make these NAD carriers, which go up to the electron transport chain and create energy. And so here's another diagram where we show in red the parts of the citric acid cycle that are actually short-circuited by propionate. So certain bacteria, such as Clostridia, can make an overabundance of propionic acid and cause this short-circuiting of the citric acid cycle and actually build up this thing called succinyl-CoA, which inhibits even more the citric acid cycle and decreases our metabolism. So there is a bit of an interesting story to this because really, this was, as I had said, a rat model that, Dr. McFabe was producing. This was back in, the late 2000s, that is about 2010, maybe 2009, 2010. I had been looking at the metabolism of kids with autism and measuring these things called fatty acids. And Dr. McFabe was doing the same thing with these rodents that were given propionic acid. And it just happened that somebody, we were invited to the same conference, and somebody put my talk right after his talk. So I had to listen to his talk, and he put up the slide of these things called acylcarnitines, which is a series of different fatty acids that are being produced in the body. And what he showed, and not to get too technical, is that certain of these fatty acids were increased in his rat model compared to the control model. And it was a very odd pattern because usually when you have a block in fatty acids, all of them are elevated, but the rats tended to have this elevation in these very short chain fatty acids and these long chain fatty acids. Well, it just happened that I was looking at kids with autism and I was measuring just metabolism, and I was presenting this data where I just happened to notice that kids with autism had this pattern, this odd pattern of acylcarnitine elevations, where there was just elevations in really short chain and long chain acyl carnitines, which matched exactly the rat model that Dr. McFabe had shown. And when we also looked at other patterns, like patterns of glutathione reduction, we found that just like the rodents, the total glutathione, which is important as an antioxidant, was very reduced in the kiddos that had this pattern of elevated acyl carnitines. So we did some other experiments too to show the parallel, but we found evidence that really these fatty acids produced by our microbiome may be actually really disturbing the way our metabolism works. And so this is kind of a summary diagram of what we think happens is that you really have your colon down here, the last part of your intestine. And many times you have good bacteria, which are in green. And so you have some bad bacteria, but you're able to balance out. And so the good bacteria is doing things like positively modulating the immune system, producing vitamins, and regulating metabolism. But sometimes, and this is what we see in many kids with autism, is we have we have a dysbiosis or many imbalanced bacteria that do bad things to our body. And we end up, instead of getting these good effects from our microbiome bacteria, we get these detrimental effects like inflammation, oxidative stress, and mitochondrial dysfunction. And so there's also some really interesting data that looked at some of the bacteria that produce propionic acid that, again, causes these autism symptoms. And this is a study done up in at Harvard and Columbia, where they looked at kids with autism and looked at the disturbances in their microbiome. And they found out that they tended to have an overrepresentation of Clostridia that produce propionic acid. And the really interesting thing is when they looked at kiddos that had GI symptoms before the onset of autism, they found out those were the kids that were the ones that had high levels of these bacteria that produce propionic acid and can cause autistic symptoms. So this kind of linked it to the fact that maybe this imbalance actually preceded the symptoms of autism and may be causative. So, how do you fix this? So, of course, we know that there is these abnormalities. So, Dr. Adams, pioneered really a treatment called microbiome transfer therapy, where he could actually replace people's microbiome and replace the microbiome of kids with autism, and see if that would actually, improve their health. And so in this study, which was on adults with autism, we had two groups. We had group A, which is what we would call the treatment group. They started out by getting an antibiotic called vancomycin, and then a clean out of their gut bacteria. So they had this antibiotic that killed most of the bad bacteria, then cleaned it out, and then they were given this microbiome transfer therapy. So essentially their microbiota was replaced with somebody's good microbiota, and they were treated here for 8 weeks with the replacing the microbiome treatment. Whereas we had group B, who did not receive the treatment. This is called the placebo group. So they received a placebo instead of an antibiotic. They had to have a clean out because it's hard to mask a clean out and pretend you're doing that. So they had to have a clean out. And then they got placebo for 8 weeks rather than the microbiome. And this is what we say phase 1, the double-blind placebo-controlled portion of the study. And then after that was done, we then looked at, well, what's the difference between continuing this treatment or starting new treatment in these individuals that received placebo. One of the things is that the second group here, in what we call the open label portion, because everybody knew what treatment they were getting, we didn't give the vancomycin to see what would happen with not giving the vancomycin. So in phase 1, people were blinded. They didn't know which treatment they were getting. Phase 2, they knew what they were getting. And of course, there's many results to this, which Dr. Adams will has presented and will present other times. But I'll give you a few cases of what I saw at my site. So we did this at 2 sites, one at ASU and one at Phoenix Children's when I was there. And so this is a 25-year-old lady with autism. She had normally developed until 18 months and then had a sudden loss of skills. She was high functioning enough to graduate with high school with a certificate. She had developed chronic constipation at 7 years of age. And then at 17, developed chronic abdominal cramping and heartburn. She also had chronic headaches and teeth grinding. Her nonverbal IQ was high average. And so we can look at her autism symptoms. So we measured the autism symptoms on something called the CARS, the Childhood Autism Rating Scale. And we can see that starting out, she was in the severe range for autism symptoms. Then as she received the treatment, or just receiving the treatment in the double-blind placebo control, she went down from high severe into the moderate range, then continued to receive treatment, went down to the middle moderate range, and then afterwards we followed her up and she continued to improve, going down to have mild symptoms. And so when we looked at her other symptoms, so one of these here, this dark blue line, are actually her GI symptoms. We can see that her GI symptoms also went down precipitously with the treatment, and the parent rating scale of how she was doing also went down significantly. One of the other participants, which is another example, was a man, a 23-year-old young man with autism who developed normally until 18 months of age and then suddenly lost skills. He developed colic at 6 weeks of age and then had chronic diarrhea starting from early in life. He had food allergies to gluten and dairy, also had an abnormal EEG and 2 seizures in his life. He was low functioning with an IQ of 50. So with treatment, we see that he's started in the severe range here in the beginning. And as treatment went on, the end of the double-blind placebo control phase 1, we see that he had symptoms that were moderate. Then at the end of the open label, it went down to minimal symptoms, and he kept improving and stayed with minimal symptoms all the way up to 18 months. We have quotes one quote from from another patient, and this is from his mother saying, "Our experience with the FMT was life-changing. Practically, our son spends significantly less time in the bathroom, and his levels of pain due to constipation has been greatly reduced. Socially, he is more aware, more connected, and has deeper friendships and a very active and happy social life. His attitude, grades, and his emotional intelligence improved. We have seen these benefits continue over the last couple of years." So he continued to improve even after the treatment. So again, we're interested in whether the mitochondria improve. So what we did is we actually took blood samples from these patients at different phases of the treatment, and we put it into this device here we call the seahorse, this fancy machine we have in our lab. No seahorses were harmed. This is a machine that measures mitochondrial function by measuring something called the respiratory rate, or how much oxygen is used up. And I won't go through all the complicated nuances here, but suffice it to say, we measure different aspects of how the mitochondria is working, including how much ATP it makes. That again is that energy molecule that's important. How much what we call proton leak, and proton leak is how it can control oxidative stress. We measure something called maximal respiratory capacity, and that's how hard the mitochondria can work if it needed to work really hard. And then something called reserve capacity, which is how much reserve the mitochondria has. And this number is very, very important, because this tells us how much the mitochondria is being stressed, and how much more stress it can take to support the body if it needed to. So we look at our results, and with this are the measurements that we saw at baseline and the end of phase 1, or the end of the double-blind placebo-controlled study. These numbers represent oxygen consumption, and you can think of higher numbers better. These curves are have to do with this certain stress test, where we stress the mitochondria to see how much stress it can take. And what we see is that from baseline to the end of phase 1, this is the individuals that received the microbiome treatment versus the ones that received placebo. I don't have here, but I can tell you for them, these lines are right on top of each other. They don't change at all. But the ones that received treatment, they were actually able to have higher proton leak to regulate oxidative stress. The maximal mobility of the mitochondria increased in this blue line, and the reserve capacity improved significantly. And the coupling efficiency, which is how efficient it is, also did, although it doesn't look impressive in this graph. We then actually divided the kiddos up into whether they were high functioning or low functioning. And we found that this effect of the microbiome on the mitochondria was particularly prominent in the high functioning kiddos. And here you can see the effect much stronger. So the red line is baseline. And then we have the end of phase 1, which is this light blue line. We can see improvements, maximal respiratory capacity. And then they continue to have microbiome treatment. And we can see the green line, their mitochondria continues to improve. And we can see for reserve capacity, the same thing, whereas the double-blind placebo-controlled study, the first treatment, we see improvement in the reserve capacity, and then we continue to treat, and we have significant improvement in the green line. And then again, in the coupling efficiency, how efficient the mitochondria went from down here about 75% up to about 85–90% in its function. So we have done other studies too, where we've actually looked at short-chain fatty acids such as butyrate on immune cells from children with autism. And we saw some very interesting facts. We divided our immune cells into those that had abnormal mitochondrial function, which is the green line, autism ADAs, and ADNs, which they had normal mitochondrial function. And what we found is that, again, you can think of, higher, better, is that you can see the green line as we give butyrate. So this graph is as we give more and more butyrate, we can see that particularly those cells that have atypical mitochondrial function, can use butyrate to actually rescue themselves and improve their mitochondrial function. And then we also found that butyrate, as you give more and more, actually improves what we call expression. That is how we use certain genes that are important in our body. We can see that giving butyrate improves genes for oxidative stress, for metabolic regulation, and for handling cellular stress. So how can we restore our microbiome to make ourselves healthier? So there's many ways of doing this. First, there's many things you can do to improve your microbiome. You can avoid mitochondrial toxins. You can avoid using antibiotics, avoid other metabolic toxins like acetaminophen. Make sure that you use good quality folate. Avoid infections, of course, and improve your psychological stress management. Improve your diet. Other things like vitamin D are very important. Sleep also affects the microbiome in many different ways. There's many different things from our environments that we get from plants and soil, air and air pollution, other medicines that we get, exposures to livestock, in our water, which is very important and influences our microbiome. We know that there's good bacteria, and we want to try and enhance our good bacteria. There's many things that we can do to enhance the growth of good bacteria and decrease the bad bacteria. We know that there's certain important bacteria that make propionate and make butyrate. And there's things that we can do to try and improve the production of good bacteria. Many of the things that will improve the growth of good bacteria includes a high fiber diet, giving prebiotics, which help good bacteria grow, and probiotics, where we can replace or enhance good bacteria in our gut. One of the other ways that are a little bit more advanced and sophisticated is that we know that certain intermediates, metabolic intermediates, help certain make certain short chain fatty acids, and so we can give these precursors to help make the right short chain fatty acids. Very important. This is most important if you're thinking about having a child and during pregnancy. So we always encourage everybody that is going to have a child to sign up at the Neurological Health Foundation for the Healthy Pregnancy Summit and the Healthy Child Guide. The Healthy Child Guide has many different suggestions of how to have a healthy child that's reduced, that will have reduced incidence and risk of having chronic disease. And across all this, I have to, recognize some of the greats in the field who have, that have been before us. Derek McFabe, who really produced this propionic acid model. Cindy Feingold, who is really a pioneer, has had a bacteria actually named after him and was a pioneer in really starting us knowing about this in autism. And of course, Emma Allen Vercoe, who's very active, and she's the Canadian microbiome chair. So the Canadians think it's so important they actually have a national chair for the microbiome. So with that, I'll thank you for your attention, and I guess take any questions if you like.
Thank you very much, Dr. Frye. So I invite everyone to go ahead and type questions into chat. And Dr. Frye, if you want to browse through there and take as many questions as you wish.
Okay, so is it possible to get you, Dr. Frye, and Adams to study my son, 24-year-old, presenting with high correlation between GI symptoms and autism symptoms? There are also other reasons to make him possibly um, very interesting and useful case. Yes, I mean, I would I'd be happy to help in any way. In our clinic, we try and do the things that are practical to or available to us, you know, in the medical field, to repair the microbiome, because it's really something that's really essential for health. Until MMT is available, um, do you recommend giving one's young adult son with autism sodium butyrate? If so, how much? So, you know, it's really something I personally have thought about a lot. Um, and from I think the problem is, and it's very interesting because there are animal studies that show that that butyrate will actually improve autism symptoms. However, for the butyrate products that are on the market, I have not seen any that really help to any significant extent. So I think it's like anything, it's not an easy fix. You know, we really have to develop a butyrate that can effectively get into the body safely, study it, understand how it does that. So we can potentially use it as a treatment. I think it's a potential treatment, but we just don't have the studies now to really use it effectively. Would you endorse the use of mitochondrial cocktails? So for every child that I see, I look for mitochondrial dysfunction and do several labs to see what the appropriate mitochondrial support is. Many children will mitochondrial support will help them, but I think it's important, at least as a doctor, to see which child that is and give them the appropriate cocktails, because there can be different cocktails depending on how metabolism is dysfunctional. Yeah, I'm happy to share my slides. Do you see a correlation between Lyme disease and autism? Really hard to say about that. How do you test for mitochondrial dysfunction? So I use both blood test, fasting blood test, that look at metabolites that may be elevated or depleted. Some of the ones that I showed you today and some of these metabolic pathways to get an idea where there may be a block or there may be some abnormality. I also use a buccal swab test that tests the some of the complexes in the electron transport chain, which also gives other pieces of information. And so there's many different flavors of mitochondrial dysfunction, so it's important to get all that information. Are there certain brands of probiotics to use and avoid? You know, I have my favorites. I'm sure Dr. Adams has his favorites. But one thing I will say that, you know, it's important to get a very high quality probiotic. So I think there's some important things. Important to get a high quality probiotic, a probiotic that is shielded so it will get down to the lower gut instead of being destroyed in the stomach. Um, in in autism, one of the things that we see that's important is lactobacillus. So I always use probiotics that have high content of lactobacillus. Um, but I think you know the jury is out. There are some studies that show that, you know, probiotics have been helpful in autism, but I think we need more studies to actually kind of nail it of what probiotic. And I think it's a little bit complicated, because what I find is that different kiddos really respond well to different probiotics. So there isn't one that's the best for everybody. And maybe someday we'll know, you know, why that is. But the microbiome is extremely complex, And it's what we talk about as a giant forest. And so you have to have all those plants. And so it's not just one bacteria, it's many of them that work together. And that's what's so intriguing about the of transplanting the microbiome, because you're not just transplanting one bacteria, but you're transplanting the forest. So it can grow and it has all its components. So it's really, I think, a good model, and I'm glad we're getting positive results. My child has 70% of mitochondria. How severe is that? He's 7 years old, nonverbal, autistic, level 3. Well, yeah, it would be good to know how you measured that, but that's a very low number that puts it in the range of mitochondrial disease. And definitely your child should have a detailed genetic workup, because we see when it's that low, many times we find a genetic cause for that. And once we know what that cause is, we're able to treat it better. Can you see out-of-state patients virtually? I do that. There's certain qualifications. They have to be seen every year in person, so it's not always virtual, but definitely follow-ups can be done virtually. Did the patients who got vancomycin in the study do better than the ones that didn't? I think probably Jim is probably best to answer that.
The answer is yes.
What, if any, is the relationship between ASD and IBD? Yeah, I think there's definitely some overlap with the microbiome and with many of the other things we see: inflammation, oxidative stress, mitochondrial dysfunction. And so many diseases, we see these same type of pathological pathways activated, and they may be the same flavor of, a different underlying pathology, but we just, not something that we have the science to understand fully yet. Does the microbiome transplant have any effect on low functioning autism? I did show a case of low functioning autism where the child got much better. And Jim, I think you would say yes, right?
Yes,
And
absolutely.
then I think your work with Pitt Hopkins, which tend to be low functioning children, show that it can even help with genetic diseases.
That's right. I just last week was speaking with a 45-year-old woman, parents of a 45-year-old woman with Pitt Hopkins. She was nonverbal up to that point, and after MTT, she was beginning to say some of her first clear words. It was so exciting to see after 45 years.
How do you start the process of determining whether a child has mitochondrial disorder or dysfunction versus disease? And that would be to go to a physician that knows how to do those tests and work through that. It's a lot of this, it's not just one test. It takes time to look at all the pieces. So really, it's good to get a doctor that understands all the nuances. What foods should we avoid that produce propionic acid? What foods produce butyrate? Really good fiber is really good to produce butyrate. Propionic acid, it's some of it's hard to avoid. It's used as a preservative in some foods, actually. So we actually do get it in some of our foods. So you have to really look closely. I'd like to know how you do a fecal transplant, oral pill or tush. The technology that Dr. Adams has developed actually is an enteric coated capsule and now some type of granules, correct?
That's right.
And so you can take those orally. Do you recommend using butyrate? Again, I don't know a butyrate that actually works well that I've seen. So I think theoretically it should, but I don't. I wish I could. I wish I had one that did work well. the hijacking of the citric acid cycle was no longer happening after fecal transplant. Again, we didn't go into that detail. We might. I think we have samples where we'll do what we call metabolomics, where we can look at this in more detail. We just know right now that overall the mitochondria was really much more robust and working much better. And so we'll have to do other studies to exactly find why. Butyrate is so hard to get in a child. Smell and taste is strong. Yeah, it depends on the butyrate supplement, but yeah, that is an issue. You're absolutely right. Recommend for good brands of probiotics, prebiotics, also might support supplements. Yeah, I mean, there's many of these. Again, it depends on your child and yourself. There's different probiotics that can help. And if you have mitochondrial problems, there's different mitochondrial supplements. There's not kind of a one size fits all, unfortunately. You can contact Dr. Frye. You can contact me on my website, Dr. Frye, MD, PhD, .com, or at the Rossignol Medical Center. When is next scheduled adult MTT study at ASU with Dr. Adams? Do you have anything planned, Jim?
We hope in a year if we can raise the 20 million we need. So we're working hard on it.
And how does a fecal transplant done? Again, it's oral through these special ways of protecting it, so it gets down to the gut. Hi, Dr. Frye. My daughter used to be a patient of yours and was going to be part of your research. At the time, we were unable to swallow pills. She can now, very interesting, become a patient again. Yeah, you can contact the Rossignol Medical Center, and yeah, they're happy to make an appointment. Our offices are up at the top, on the top of the 51 on Union Hills Road. So yeah, we'd love to see you. Hi, when is the FMT treatment going to be available for all adults? Again, Dr. Adams is working hard to get the studies going and get it approved by the FDA so everybody can have it. Does MTT help with apraxia? It very well might. I don't think that's something we measured in any of the trials, so it's hard to say, but it does seem to improve many symptoms. What is your thoughts on the MCT keto diet to help with butyrate decrease, that reduce carbon? Yeah, I mean, I think that there's a lot of the keto diet has a lot of really positive effects, and it does seem to, with the data we have so far, seem to have a positive effect on autism symptoms, on metabolism, so it's definitely an alternative, diet that may be good, but it's also important to do it under the guidance of a dietitian because there's many metabolic changes that need to be monitored. How reliably do you feel personally prebiotics, probiotics through fecal assessment and food recommendations the company Visbiome or Tiny Health are? I think all of these things and all medical tests, you should look at as a guide, not an absolute. So I think every tool you have is kind of a guide to help, and then you can do treatments, see if they help. Sometimes they help, sometimes they don't. It's not kind of a one size fits all thing. Yeah, it's unfortunately not something that's easy to fix. So I know there's a lot of ways that we're trying to do it, but many times you have to try many different ways. Does mitochondrial support help low functioning autistic adults? I've seen that it does. What about mitochondrial membrane support supplements? How do you decide if genes non-invasive cocktail? Yeah, I mean those are the mitochondrial membrane support. Yeah, some of those are interesting. Additional things to put on after mitochondrial cocktail. So the important thing is to get your basic mitochondrial cocktail down, and get diet down, sleep, other things to improve the body. And then you can try and put some of these other things that we don't, have a lot of data on, but potentially, are very good to see if they help the patient. And so that's something definitely, I think, after you have the basics down, something that you try. If it works, you continue it. If not, you move on. So as long as it's safe, there's, really no downside to trying these things. Is there a difference between using a capsule versus powder? It depends on the powder and capsule. Again, it's best if it's coated so it gets down to the lower gut. Usually the powders might not be, but the granules a lot of times are. Again, out-of-state patients can do testing virtually. In the short term, to verify changes caused by MTT, would it be better to study metabolism or microbiome? Do all Clostridia produce propionic acid? So it's complicated. So I always think it's important to look at both metabolism and microbiome. And no, not all Clostridia produce propionic acid. There's actually good Clostridia and not so good Clostridia. And so it is very complicated. Are seizures a major sign of mitochondrial dysfunction? They can. Mitochondrial disease, particularly, there's an increased risk of seizures. But there's other things that can cause seizures too. So it's important to look at many different things. I've been following Dr. Sasha Gomik's theory that a 3-month course of B vitamins along with good level of D gets body to naturally grow back the microbiome in a correct balance. I don't know. I mean, I think it could definitely help. It probably depends on how depleted your microbiome is. I would think. I don't think it would work for everybody. And if it works in 3 months, yeah, that's really terrific. But I think it's good to try, and if you get great results in 3 months, sometimes we have, kiddos really take off. That's not the usual case, but if they do, then, we consider, taking them off certain supplements if they don't need it. But that's not usually the case that I've found. Are your results published? So they're they're in the process of being published. It's unfortunately not so easy to analyze a large amount of data and write the papers. So we're trying our best. Hey there, Dr. Frye. Told triplets here. We miss you. Well, thank you. I miss you guys too. The triplet sister is on some mito support still, but suffers chronic constipation, recent diagnosis of POTS. Any correlation with mito and POTS? Yeah, most definitely we see POTS along with autonomic dysfunction as a consequence of mito. And I hope you guys are doing well otherwise. Why do probiotics sometimes cause constipation? Again, not all probiotics are for everybody. It's a very complicated forest in there, and if you give the wrong plants, sometimes it doesn't play right with the rest of the forest. Can out-of-state testing patients do virtually? Yes. Can any bacteriophages against Clostridia? Definitely, I don't think in the clinical world, maybe some in the research world, but nothing, I think, available for clinical use. My child has never said any functional words, just babbles and doesn't make much of an effort to say any words. I don't know what to make of it. She never had any words as a baby, that she regressed and lost. What do you think can be the cause? There's a wide number of different causes, and that's what we do in the clinic, is actually take a wide look at all different causes so that we can see what's most appropriate to treat. A lot of times there's not just one cause, it's a combination of things. So we have to kind of hit things from multiple angles. But definitely is something that is worth doing. Our girls have SHANK3 mutation, previous tested, did not identify mitochondrial disorder dysfunction. As they age, is this something that can change? Yeah, and we think that mitochondrial dysfunction is part of aging, and especially in kiddos with neurodevelopmental disorders. So it's something that can, I think, develop over time. After doing biome site test, my daughter's acetylcholine is 0. What do you think is the cause in kids with autism? There's probably many different causes, so it'd be important to look at the reason why that might occurring. There's many different avenues to the same thing. What do you think of Namenda medication for kids with autism? I use it all the time. It's very helpful for some kids, and others it doesn't work. So again, you have to try things on the appropriate child. It's not my first thing to do. It's usually used in more refractory cases. In your clinical practice, does children improve dramatically with mito treatment? Sometimes, sometimes not. Yeah, sometimes it's a little bit harder than that. Can you elaborate why recommending no acetaminophen? Acetaminophen depletes glutathione, and glutathione is already depleted in kids with autism, and it's also a mitochondrial toxin, and the mitochondria isn't working well in kids with autism. So in those that have a predisposition to have low glutathione or mitochondrial dysfunction, I recommend staying away from acetaminophen usually. When do you think MTT will be available in the US? That's a question for Dr. Adams.
We hope to have it approved for Pitt Hopkins and on the market in 2 years.
Wow, that's really great. What is the difference between mitochondrial dysfunction and oxidative stress? They're two related things, but separate. Oxidative stress is essentially free radicals that damage things in the body, proteins, DNA, and lipid membranes. The mitochondria, as you saw, is very complex organelle, and so it can be dysfunctional for many reasons. Oxidative stress can damage much of the mitochondria, so that's why we look at how those things play together. When the mitochondria doesn't work well, it can produce more oxidative stress. So those things can play off each other. My 7-year-old is on Mito cocktail, and each mitoswarms come back with lower numbers. What are other testing should be done? Definitely, I don't rely on one test, and that's why usually I do some fasting blood labs, also to see what is happening metabolically. So that's always important. If things are getting worse over time, it's very important to look into that, because there might be more of a mitochondrial disease where the mitochondria might actually be getting worse. And if that's the case, then you might need to be more aggressive. Or it might just be variation in the test. So, but it's important to take that seriously. The psyllium fiber capsules help. Fiber is always good, and tends to improve good fermenting bacteria. Dr. Frye, are you taking new patients? My son is 24. Yes, I am. Will there be a replay of the webinar?
We're recording the webinar, and we'll send a copy of the webinar. And Dr. Frye, I think you said also a copy of your PowerPoint.
Yeah, you're happy
to everyone who registered.
How do I find a doctor with my area of Virginia that tests for gut dysbiosis and mitochondrial disorder? You can definitely contact me. You can look on the MAPS website, the Medical Academy of Pediatric with Special Needs. People that are fellows tend to be, know some of this. So, but of course, it's important to, each doctor has their strengths and weaknesses. So it's important to, see what their strength is and if they do that work. What about butyrate suppositories? Yeah, again, not a lot of evidence, there, but butyrate should theoretically work and should theoretically work with suppositories too, but there's no evidence of that. So I don't, it's not my recommendation to try something that we don't know much about. Same question as one above, 35-year-old low functioning with seizures in San Diego. Sure, yeah, definitely. We see patients. There's a Rossignol Center down in Southern California too, which you're welcome to go to. Dr. Adams, are you still in a trial stage? How can we go about getting MTT? My son is 7. That's a question for you, Jim.
Yeah, again, we hope to open another study for children and another study for adults in a year from now. If we can raise the next 20 million that we need, these will be much larger studies. And then we hope to have it FDA approved in about 2 years for Pitt Hopkins. Dr. Frye, you've been very gracious with your time. We've been taking Q&A for 30 minutes. Maybe take a couple more and then close it for the evening.
Okay. Yeah, I think we'll put some contact information on the website with a PowerPoint so you can have that. Carnivore diet. I think it's probably good to have some fiber in your diet, not just all meat. But I contact a nutritionist about having a balanced diet. Usually balanced organic diet is good, of course, for any meats. It's all good to have grass-fed organic meats, but that's not the only thing I would eat. Constipation appears to be a big issue in ASD. Could you speak to MTT terms? Talk about specifically the low MTT related factors like redundant colon play. I know definitely constipation improved in many of our patients. Also suggests for love bombing with FDA with support to approve a C-section on investing sites. Yeah, I mean, the FDA, from my experience, is very receptive. They're there just to make sure that the products that are out there are safe. So from my experience, they're very receptive to these new products. So I don't know if love bombing them is going to help as much. I think they're pretty much on the right team. I don't know. What's your opinion, Jim?
The FDA has been very helpful to us. It's just that they have high standards. And so we need to do large studies, larger than we've done. And for that, we just need more funds to do those studies. But the FDA has been very helpful to us. They granted us both orphan drug designation and pediatric rare disease designation for Pitt Hopkins. That makes it much easier to get drug approval.
Yeah, I mean, that's one thing I think a lot of people don't understand. The FDA doesn't fund any studies, so they're happy to help you in any way they can, but they're not there to fund your studies. So I think they do the best with what they have. So I think we'll leave it there. I thank everybody for coming. It's great that you have all this interest. It's great to see everybody. So yeah, take care and thank you.
Thank you very much, Dr. Frye. Great presentation. Very much appreciated.
Well, thank you guys.